What is this? How did I get here?
Wait, who are you? Hello? Help! Help!
Relax, everything's alright. You're in a hospital bed about to receive a surgery. The reason why you can't remember anything is because you were given a heavy anesthesia for the operation.
What? Okay, that makes sense. But what surgery am I getting? Did I sign up for it?
Yes, you signed up for it when you were conscious. It's quite a nice surgery.
Well, what is it?
You're getting a green fluorescent protein gene insertion.
A what now?
Basically, we're inserting the green fluorescent protein gene into your DNA that allows you to code for the GFP protein, making you shine.
Yeah, I'm still not getting it. Is there any way you could break it down further?
Well, as a doctor, it's part of my contract to tell you everything if you become conscious. So sure. Let's break it down into really simple terms. The kind of surgery you are receiving is one that changes your DNA. This is called a mutation.
Yeah, I've heard of those before. But won't that make me like a monster with goo coming out of my body?
No. Not all mutations are harmful like that. Some mutations can be neutral, and a lot are actually good.
Why's that?
Well, they can increase genetic diversity in a species and introduce new beneficial traits.
Right, so what's the mutation I'm getting?
We'll get there soon. What you are is a species of fish called a daimyo, and a lot of humans like to keep you as a pet.
So what kind of mutation would be beneficial for me then? I mean, if I'm being fed by a human, I don't really need any new traits to survive.
Well, let's think for a bit. What are some things that can make you more appealing to a human?
Well, humans like colors.
Exactly. That's the kind of surgery you're gonna get.
I get more colorful? But how?
Through the green fluorescent protein. It's a type of protein that absorbs blue light and reflects green light. So anything that possesses the protein will appear a vibrant green.
But how do I continue to glow once I have the proteins? Won't they wear off?
You're right, they will. That's why the surgery you're getting doesn't give you the protein. It gives you the ability to create the protein so you will glow constantly.
Well, how does it do that?
Jeez, you really do ask a lot of questions.
Hey, you said you're required to answer anything I ask.
Yeah, you're right. It's just usually my patients aren't this needy.
What did you just
Okay,
say?
so the green fluorescent protein gene, or we'll call it GFP from now on, is inserted into your DNA through a technology called CRISPR, which essentially allows scientists to delete a certain part of DNA and replace it with whatever they want, acting as a DNA polymerase.
So how does CRISPR work?
You know what? I can't do this anymore. You're bothering me. You thought I was just a normal doctor? Well, I'm a magical doctor too. How else do you think I perform all these surgeries?
What's happening to me? What are you doing?
I'm shrinking down your consciousness further than you could ever imagine and placing it inside of your normal-sized body. You'll have to act as the DNA polymerase protein and create the mutation yourself. So the surgery yourself.
What? This is so unfair.
It should teach you a little bit before annoying me next time. You should be the right size by now, although I couldn't tell. Walk around, get comfortable. You should see some nucleotides floating around. These are what code for your DNA.
These colorful things?
Yeah, exactly. Now you see that strange thing off in the distance? Walk towards it. That's a segment of your DNA, a gene. It just so happens that this is one that we have to alter.
Well, that's convenient.
It sure is. Now there will be a guide RNA molecule coming down soon, and it will guide an enzyme called Cas9 to that gene. It cuts out part of the gene that we don't need and allows scientists, or in this case, you, to insert anything else you want. This is where scientists insert the GFP gene.
Okay, now what do I do?
Now it's time for you to start adding your mutation. Pick up that A base pair and add its phosphate region to the 3' end of the sugar.
The what?
Count the carbons on the sugar and add a phosphate to the 3rd carbon.
Okay, there we go. It's in place.
Great, now do this again with another A, then another A, then a G, A, G, C.
How long is this going to take exactly?
Well, you've gotten one already, so you'll need exactly 62,387 more. And an A. Perfect.
Great, now am I done?
You only did the 5' to 3' direction. DNA molecules are anti-parallel, so they have another strand running the opposite way. Now you have to do the 3' to 5' end.
Not again. Why?
That's the last one. Now those ligase molecules will glue together the Okazaki fragments you just made. Since we added so many bases in the DNA, the type of mutation you just made is a series of point mutations, which is a mutation that happens to one specific base. The mutation consists of insertion and deletion. Great work. You successfully mutated your body.
Hooray! Can I come back now?
Not so fast. See, we're only halfway there. What you've done now is essentially made a cookbook for your cell to refer to. You've only made the mutation. Now you have to make the actual GFP molecule.
But couldn't my body just do that now? Why do I have to do it?
Look, you're lucky I'm only making you do this once. I could have you do it forever. So now that the DNA is mutated, you're going to act as a molecule called the messenger RNA.
What does that do?
It's very similar to DNA. It creates an RNA molecule based on the base pairs in your DNA segment. This RNA molecule later codes for proteins like GFP.
Oh, I'm getting close then.
Not quite. Now you need to create an entire RNA molecule based on the DNA segment you added in. This process is called transcription. It's basically like taking that cookbook recipe and rewriting it onto an index card so you can bring it to your kitchen.
Are you kidding me? How do I do that? They're all stuck together.
You'll be able to in a second.
Whoa, what was that?
That was a helicase molecule. It breaks apart the hydrogen bonds in DNA, or essentially unzips the two strands. Now you can go in and add the corresponding base pairs to form your mRNA molecule. When you're adding the base pairs, make sure to replace the T with U instead.
Yay, my favorite job. Okay, great. It's honestly getting easier.
Seems like it. Now that you have the recipe on an index card, you can finally bring it to the kitchen.
What's the kitchen?
The kitchen is a thing called a ribosome, the site of protein synthesis. Go follow that mRNA molecule out of your nucleus and over to the kitchen.
Whoa, this is incredible.
Don't get too caught up now, you still have to do the process of translation.
Okay, explain.
Translation is basically the process of using the index card to assemble ingredients and turn them into your cake. In this case, the ingredients are called amino acids and the cake is your protein.
You mean GFP?
Bingo. To do that, you need to act as a different type of RNA. Transfer RNA. There should be a few floating around you.
Yeah, I see one. It looks very complex.
Indeed it is. All you really need to know is the bottom, which is the anticodon, meaning it is the opposite of the codon it attaches to, and the top, which grabs an amino acid.
So the amino acids form together to build a protein.
Exactly. Now read those first three bases. What do they say?
AUG?
Great. These three bases are one codon. What's the anticodon for AUG?
UAC, right?
Correct. Now, the codon AUG codes for the methionine amino acid. Go find one and bring it back here.
Here you go.
Good. Once you bring it to the ribosome, the translation process begins. Think of the ribosome as the baker. It reads the index card and assembles the ingredients in a specific order to form the cake. What does the next codon read?
UUU, so my anticodon is AAA.
Right, so UUU codes for phenylalanine. Go find one and bring it back.
Do I put it next to the methionine?
Yep, put it into place and a peptide bond will form between them, linking them together.
Please don't tell me I have to do this with every single codon.
I hate to break the bad news, but yeah.
That better be the last time I have to see a U, A, G, or C again.
Don't worry, you finished. Now the peptide chain will disconnect from the ribosome and will fold itself to form a protein, your very own GFP. Now that was just one. Try doing this across millions of cells, each of themselves creating up to millions of GFP proteins every single day. You are utterly insignificant.
Gee, thanks. Alright, I did your work. Can I be brought back now?
Fine, I guess you've earned it. I've got a surprise for you once you come back.
Okay, I'm here. What's the surprise? Ew, what is that? Why is it so green? Wait, I'm green too.
Congratulations, you've made your own GFP cake, and now your cells are too.
Thank you, doctor. I'll always remember this.